All right, we'll get started so we can stay on time here. Welcome to this week's EBRC virtual seminar. I'm Becky McElprang, a postdoc at the EBRC, and I'll be hosting this session. Before we get to our speakers, note that you can find the remaining seminars in our series by going to our website, ebrc.org. Also, we're recording some of the seminars, including the ones today, and posting them to our YouTube channel. The first few talks were just uploaded earlier today, so you can search for EBRC on YouTube or just visit our seminar page on the website for links. Additionally, for graduate students and postdocs, both of our speakers have agreed to stay on the line for a bit after their talks are finished to speak with students and postdocs who want the opportunity to ask more questions about research or their career paths. So with that, let's get started. We'll hear first from Dr. George Liu of Rice University, and then Dr. Jared DaCosta, who is a research chemist with the U.S. Army CCDC Chemical Biological Center. Each speaker will have 25 minutes, followed by a 5-minute Q&A. And during the Q&As, feel free to ask questions via the chat feature or using the raise hand feature on Zoom. Okay, so Dr. George Liu obtained his undergraduate degree at the University of Alberta in Canada before moving to the University of California, San Diego for his PhD study on structural biology. He then became an NIH K99 postdoctoral fellow in the lab of Mikhail Shapiro at Caltech, where he focused on the development of non-invasive technologies for biological imaging and cellular control. His independent research group was launched in the bioengineering department at Rice University in 2020, or earlier just this year. His work on acoustically erasable imaging agents has been recognized by the Young Investigator of the Year Award from the World Molecular Imaging Society. You can visit his website, liulab.rice.edu. With that, I'll turn it over to Dr. Liu. Go ahead and share yours. Let's see, I'll stop my share. Go ahead and share your screen.
All right, can everybody see this? Yeah, okay. Thanks everyone for coming to here. It's really great to see such a turnout, and also thanks the organizer for the opportunity and organizing this wonderful event. I think it's really nice that we can all sit in our house and probably share a bit of our research. So let me turn on the laser point. Ooh. Okay, so as Becky mentioned, so my lab recently started at Rice University. So what I'm going to share with you today is mostly the 2 stories that I've been working on during my postdoctoral time. And at the end, I'm going to talk a little bit about the future direction that we are going to pursue in our independent group here. So this 2 story all concerns with the engineering of a group of protein nanostructure called gas vesicle, and it's applied to magnetic resonance imaging and optical coherent tomography. So talking about imaging modality, often the first thing that comes into your mind is probably fluorescent protein, right? So this started with green fluorescent protein, later on got expanded into a whole range of different multicolor fluorescent protein that really revolutionized the whole biomedical research field. So for this crowd here, who are people that thinking about engineering biology, I think green fluorescent protein are really familiar to you guys. In fact, if you pull out the two papers that really founded the whole field of synthetic biology, look at the figure 1A of these 2 papers. What is this and what is that? In fact, they are both green fluorescence protein. In fact, the majority of the data in those 2 papers are from green fluorescence protein. It can be really, you can argue that without fluorescence protein, perhaps the whole field of synthetic biology is going to be pushed back by quite a few years. So now, of course, fluorescent protein has been developed for several decades already. Now biomedical research, it really expanded tremendously in both the spatial and temporal scale. So fluorescent protein are not able to cope with all the current need of biological research. One of the fundamental limitations for fluorescence imaging is the imaging depth. How far can the light actually penetrate into biological tissue. As we know, that usual method to look at fluorescent proteins, such as confocal microscopy, can only penetrate a few hundred micron into biological tissues. So this works great when you look at transparent organisms such as worms or zebrafish. It faces a challenge when you start looking at a mouse or eventually into human beings. So this is especially important when you are thinking about cell-based therapy that a lot of us are working on and towards in the next few decades. There, we right now don't really have a way of tracking down all the cells when you perfuse it back to human patient. And we really need a way to do that non-invasively. We can't really implant the optic fiber to do it. So how do we tackle this question of addressing this additional tissue penetration depth here? So the strategy that we are thinking about is actually utilizing this other imaging modality, here illustrated optical coherent tomography, ultrasound, or MRI. These are actually imaging modality that's already well established in the clinics. They can produce robust image and has been used in human beings. However, they traditionally have been used to look at tissue morphology mostly. They haven't been used in the same way as confocal microscopy to look at, for example, gene expression or molecular details. The reason that is the case is because there isn't an equivalent of green fluorescence protein that can communicate with this other imaging modality. So that this is really our target. Can we actually engineer something that functions just the same as green fluorescence protein, that are genetically encoded, made of protein, can be expressed inside the cell, but are able to communicate with, say, sound wave or magnetic field? So that might sound like a daunting task, and that actually leads us to look at really special type of proteins. So one group that we are working on is so-called gas vesicle. So if you fly through the South San Francisco Bay, you will actually see this beautiful salt pond. One type of gas vesicle is isolated from photosynthetic microbe that live in this salt pond. So for this microbe, they need to accomplish a daily task. They need to regulate their cellular buoyancy because these are photosynthetic microbes. They need to compete for the maximal amount of sunshine by floating to the very top surface of the water, right? So they actually evolved this tiny gas field protein nanostructure inside the cell to help them to float. If you zoom in one of them, they are usually a few hundred nanometer in size. They are made of entirely proteins. So the name vesicle is really a misnomer for historical reason. They don't contain any lipid. In this case, the protein form this 2 nanometer shell that allow air to freely exchange in and out, but are able to prevent water from condensing inside. So that's how they actually keep the inside of gas field and keep inside dry. We have already identified dozens of photosynthetic microbes that contain the gas vesicle. So often you will see a gene cluster that actually encode the gas vesicle. Only 1 or 2 of them are actually the structural component that form the gas vesicle. It often comes with a set of other accessory proteins that are essential for its assembly process. However, right now we actually don't understand this whole assembly process much. We don't know the function of a lot of these genes. So this is going to be one of the key research directions in the next few years. Okay, so this unique gas compartment, back to the imaging, this unique gas compartment actually presented a very interesting material property, right? So usually for biological tissues, other than lung, are all filled with waters. So the material property of biological tissue are more or less the same as pure water. Gas in this case presented dramatically different type of material. So what matters for ultrasound is this property called acoustic impedance, which depends on the density of the material as well as the speed of sound in the material. So in this case, gas and water really differed by several order of magnitude. So what that means is when a sound wave actually propagates to this gas-water interface, it will strongly scatter back. And that's how gas vesicle can actually light themselves up in the ultrasound imaging. So we did that we will show that if you configure bacteria to express gas vesicle inside the colon, then you could actually see them with ultrasound imaging. So this is our work led by my colleague in my postdoctoral group in Mikhail Shapiro groups back at Caltech. I was really inspired by thinking more into material property. This material property of gas really presented a lot of opportunity. So I was exploring other material property. I've come to this property called magnetic susceptibility, which is actually something detectable by magnetic resonance imaging. In this case, again, gas actually shifted quite a bit far from water. So although it's not several other magnitude, but in fact that all the aqueous tissue are right around -9. So if you look at the noise, signal to noise level, it was actually quite good. So, and that's one of the main stories that I'm going to share today. And I'm also going to briefly talk about this other material property called refractive index, which actually is the underlying mechanism for gas vesicle to light up in the optical coherent tomography. So as some of you might know, that MRI are a method that look at nuclear spin of water. So they don't directly look at the gas themselves, all right? So how does it work with to image a gas vesicle in this case? Well, we did a simulation. If you place a gas vesicle inside a strong magnetic field, which is the case in MRI, it will actually create this magnetic field gradient right around it. So now when a water molecule goes through this magnetic field gradient, it will experience an enhanced decay of their signal, which is actually the property that MRI measures, all right? So it's not directly measuring the gas component, but measuring the relaxation property of water molecule. So how does it really work in practice? The first experiment we did is to constitute a concentration series of gas vesicle, and actually checked it with 3 different types of MRI imaging modality. In this case, these are just jargon from MRI, quantitative susceptibility mapping, T2 star and T2. But really the bottom line is, as you increase the concentration of gas vesicle, you see an increasing amount of contrast or decreasing amount of contrast. All right. So as I mentioned, gas vesicle is genetically encoded. So as a proof of concept experiment, we configured E. coli to express gas vesicle under the chemical stimulus IPTG. And in this case, these are also a phantom just as the one above, but in all the wells here are no longer purified gas vesicle, but actually cells that are configured to express gas vesicle on the IPTG or express other proteins. So in this case, the control sample is expressing green fluorescence protein. It doesn't really show up in the MRI image. Also, bacteria that contain the gene but are not induced to express will not show up in the MRI. So that's actually about, I would say about maybe a third of that whole story. That shows that, you know, gas vesicle can be a contrast agent for MRI. But what's most exciting about this type of contrast agent compared to other MRI contrast agent is that they can enable background-free imaging. All right, so first a little bit background that why do we want background-free imaging? MRI is a great imaging modality because of their soft tissue contrast. So often we use MRI to look at the brain, look at tissues that tells you details about blood vessels, tells you where the boundary of tissue is. However, this great soft tissue contrast become a double-edged sword when it comes to the development of a contrast agent. Because often when a clinician look at a black spot on an MRI, it can be either the endogenous tissue contrast or the contrast agent themselves. So often you need to design contrast agent to be bright enough to overcome the endogenous tissue contrast. It would be great if we actually have a way to in situ turn on and off the contrast agent. So here actually gas vesicle offer a way to turn themselves off. So this is actually based on their acoustic property. So we know that when we deliver a pressure wave, can be in the form by ultrasound transducer, then it can irreversibly collapse gas vesicle. So in this case, showing in the electron microscopy imaging, you can actually squeeze out all the air, leaving the protein shell in there. But because the contrast depends on the air, by doing so, we hypothesize that you will be able to erase all the MRI contrast. So the first experiment we did is, again, using purified gas vessel inside the phantom. As you can see, after the MRI, after the ultrasound, the intensity of the well essentially turned back to that of a blank buffer. So now you can do a simple image subtraction, and now the image will be freed from all the background contrast, but only pertain the intensity that's specific to gas vessel. So we did that also in vivo setting. In this case, injected the gas vesicle inside the brain of a live mouse and show the same paradigm of obtaining a background-free image. The next task, we are inspired by the trajectory of fluorescent protein. So when the green fluorescent protein was first engineered as a contrast agent, one of the immediate next question is, can you actually do multicolor imaging? So what that means is, can you actually track down different genotype of fluorescent protein in one set of image, right? So that's important for tracking multiple group of cells or monitoring several genes of interest. Can we actually do the same for gas vesicle on the ultrasound MRI? Well, in this case, is different. So fluorescence imaging can be done because it comes with a range of different optical property. But for ultrasound MRI, they are both monocolor intrinsically. So how do we differentiate different type of gas vesicle? In this case, we actually differentiate based on, based on their rigidity. Turns out that based on the different genotype of gas vesicle, they will have their characteristic collapse pressure. So here is experiment where you ramp up the hydrostatic pressure, you will see the weaker one get collapsed first before the stronger one. So now we can have a way to differentiate the two by taking an image first of the total population and then design a pulse that selectively collapses the weaker one and take another image and again do image subtraction. So that's exactly what we did here with two purified samples as well as a mixture of the sample. Now we can do image subtraction, render it with pseudo color, and you can achieve a pseudo multicolor image. We're also wondering if you can actually engineering a sensor for some biological function. So what I mean by that is, can you actually turn the contrast up and down in response to certain biological stimuli, such as neurotransmitter or protease, all right? So here we are asking a little bit more than just using ultrasound to completely erase the gas vesicle. We want to turn it up and down. So how do we, can we actually have any way to do that? Well, one strategy is coming from thinking more into the MRI principles, why the gas vesicle actually has the contrast. So in this case, this is a plot of the T2 and T2 star mode of the MRI contrast. The relaxation rate, which is what you measure with MRI, has to do with this delta omega R times tau D. So this is the, I'm going to bring back the simulation to tell you what these two things means. So omega R is the frequency shift in the field gradient I just showed. So gas vesicle will produce a field gradient right around it. The frequency is really the difference from one end of the field gradient to the other, which in this case depends on the susceptibility of the gas versus water, which is something we really can't change, all right? So what we could change is probably the water diffusion time. In this case, it's the time for water on average to diffuse through this field gradients. So if we can make the gas vesicle all cluster together, we can significantly increase the size of this field gradient, as well as creating a lot of channels in between gas vesicles. So we hypothesize that will actually significantly change the water diffusion time and therefore turning the contrast up and down. In fact, with the unclustered one, we calculated it will be right around here. With cluster one, we will be able to push them into more of this so-called intermediate regime for MRI. So we did a proof of concept experiment using biotinolated gas vesicle and clustered them with scrubbed evidence. We showed with the BLS and electron microscopy that we can cluster them. And when it comes to the MRI imaging, here on the left is two control samples that we either omit biotin or omit scrubbed evidence. And then the last sample is when you actually cluster them together. We can see a dramatic difference in contrast for the T2 star and T2. Okay, in this case, actually T2 star differ by more than 10, by more than a factor of 10. So we're really happy with the amount of contrast we can change. Interestingly, for QSM, which is the way that spit out this susceptibility value, it doesn't really change much. So actually, in this case, it quantify the overall susceptibility in the sample, which depends on the overall quantity of air in this case. And in this case, because we're just modulating the clustering, we're not changing the number of gas vesicles. So the total susceptibility actually didn't change. So this is great that we can now use QSM to perhaps monitor the total concentration of gas vesicle, and then using T2, T2 star to monitoring their clustering state. So this gives us both information. So I'm going to switch gear to briefly talk about optical coherent tomography, which is a more recent stories. So for people that perhaps are not quite familiar with OCT, so OCT is actually a pretty common imaging modality. If you go to the eye doctors, so in the ophthalmology clinics, OCT is really the go-to method to look at your retinal anatomy. So a lot of you probably have seen this kind of image. And more recently, actually, OCT has been used in a wider variety of medical settings as well as industrial inspection. So a lot of the recent work you've seen in intravascular OCT or endoscopic OCT. Also, OCT can be used for image biofilm because it offers a intermediate penetration depth as well as the field of coverage that really are ideal to encompass the whole biofilm. So in a nutshell, the way that OCT works is by sending a package of light, all right? And it will bounce off some biological scatter. So traditionally, this is small organelles or membranes of the cells. And based on the time of sending the pulse, the time in echo back, you can use the time of flight information to pinpoint how deep the scatter is. And that's why you call it the tomography method, because you can now figure out both in the 2D dimension, but also in the third dimension, how deep they are. The intensity of OCT in this case, as you can see, depends on the scattering property of the molecules. So that translates to refractive index. In this case, actually gas also presented a very strong reflecting surface for the OCT imaging agent. So to check if it works, the first experiment we did also is to have a concentration series of gas vesicle. In this case, we actually push it down to tens of picomolar in concentration. And we can readily observe the gas vesicle intensity. We can do similar things as using ultrasound to actually wipe out all the intensity from the gas vesicle and actually do image subtraction to map out the intensity that's specific to gas vesicle. We collaborated with clinicians at UC San Diego because OCT has been often used in ophthalmology settings. So we want to check that how does the gas vesicle work in the mouse retina. In this case, we used two clinically proven injection route into the eye of the mouse. So subretinal injection as well as intravitreal injection, and used the FDA approved imaging modality to look at the presence of gas vesicles. So right here, this is a layer of retinal, and right above it, all the white contrast is actually from injected gas vesicles. So we could also do in situ ultrasound erasing. So in this case, actually using a portable ultrasound transducer from Philips Lumify, we are able to deliver enough pulse to actually erase the gas vesicles. So showing here, after erasing, you can actually remove all these white signals. So now this is actually a way to confirm that, yes, all those signals are actually coming from gas vesicle. But retinal, you basically can see is still the same signals on the OCT image. So due to the time limitation, thanks for the reminding of the time. So I want to wrap it up. So hopefully today I briefly talked to you about that gas vesicle are a genetically encoded contrast agent really for a wide variety of imaging modalities. So looking into the future, I think this is, on one hand, this is really a time for us to actually go back to look at this group of molecule a bit further, right? So a big direction that my lab is pursuing is really to understand on the mechanistic level, on the structural level, how this gas vesicle actually gets manufactured, how does it get assembled. And from those information, we want to understand and further come up with new ways of designing novel variants of gas vesicle that can give you enhanced imaging capability, as well as some other applications that we are pursuing. So that's one of the main directions in my ongoing group. And on the other hand, the application of gas vesicle are really not just limited to the 3 imaging modality I'm showing here. All right, so my lab is also actively pursuing directions that are outside of imaging field, especially in terms of controlling cellular signals and chemistry. So just as a teaser of this direction, I want to give a showcase of 2 works that are actually, again, led by my colleagues back in Mikhail Shapiro group, but are recently published in the last few months. So one is actually using gas vesicle, in this case, use this way, so-called acoustic trapping, to spatially control the location of gas vesicle. So acoustic trapping is similar to optic tweezer, perhaps you have heard about that, but this is the version in ultrasound. So here we are actually able to show that both with purified gas vesicle as well as with cells that express gas vesicle, you can use focused ultrasound to move them in space. On the other direction, we're also exploring this cavitation effect, which is actually nucleating a lot of air that can be released by gas vesicle, and designing an ultrasound pulse scheme that can trigger a violent explosion to release the cellular content. So this can be useful if you actually configure the cells to express certain payload or drugs. And then you can actually use focused ultrasound to control their release in situ at the specific locations. So this just gives you a showcase of gas vesicle are really not just limited to imaging application. We're really excited in the next few years to explore other directions, especially in terms of cellular control. So with that, I would like to thank my postdoctoral mentors, because most of the work I'm sharing today are from my time back at Caltech, as well as the co-authors and also the wonderful collaborator that helped out in the 2 stories I shared with you, the MRI and OCT. And here I just opened my lab earlier this year. So luckily I arrived Houston before the whole viruses hitting, and we are actually actively recruiting grad student and postdoc at this moment. So I'm happy to talk with you guys afterwards. And lastly, I would like also thanks my wife and the wonderful kids that comes with me and be supporting my research. So with that, I'm welcoming any questions.
Thank you. That was a great talk.